Hello everyone, I'm Matteo Bernardello and I'm currently PhD student at ICFO, a research institute based in Castelldefels, Spain. In this lecture, I will give you an overview on microscopy. Microscopy is a broad field that, in a very general sense, aims to permit the visualization of objects that are too small to be seen at the naked eye. In this course, we will focus on light microscopy, which refers to the subset of microscopy techniques that use optical elements and the different properties of light to generate a magnified image of the sample of interest, from which will then be possible to extract useful information. Particularly, we want to describe its application in biological study, in which the sample of interest are cells, tissue, or even entire organisms and their biological processes that have been introduced in previous lectures. As there is a big variety of microscopy techniques, it is important to understand which microscope will give better result for a particular biological application. To do that, let's first define some basic parameters that will help in understanding the performance of a microscope. So, one of the most important parameters in microscopy is the spatial resolution. Let's suppose that our sample only consists of two particles, ideally 2 points. As I've been explaining in the previous lecture, those 2 points will be imaged as the PSF dictate, broadening their size on the final image. Our aim is to be able to distinguish them as they approach to each other. This capability depends on the actual distance between the particles and on the performances of our microscope. As an example, from the first row, we can clearly say that there are 2 different objects. As those particles approach each other, however, it becomes more and more difficult to distinguish them in the final image, until we cannot anymore understand if there are 2 particles or a single particle. Therefore, the minimum distance at which 2 objects can be close to each other and still be discernible in the final image is the spatial resolution. Another important parameter in microscopy is the temporal resolution. Let's suppose now that we want to follow the path of a single particle. We can take an image at the time t1 and then repeat the imaging after 1 second at the time t2. We can now have the position of the particle in those 2 time points, and we could suppose which path it followed from the first to the second position. However, a number of paths are possible, and we cannot conclude on any of them as we miss the images of the particle in between the 2 points. To solve this problem, we could repeat the imaging processing, but instead of taking 1 image every second, we decide to take 1 image every millisecond. In this way, we can better follow the particle and have a better idea on the path that it takes. So the ability to follow this particle, or in general to follow an event, is depending on the speed of that particular event and on the capability of our microscope. The minimum detectable time interval between 2 events is the temporal resolution of our system. The last parameter is the field of view, and it basically defines the portion of the sample that we want to image. As an example, if our aim is to study a single cell, a small field of view of around 60 micrometers would be enough, as the cells are smaller than this range. However, if we are interested in visualizing a big portion of an organism, as an example the zebrafish head, we will need a field of view of at least 700 micrometers, so that our specimen entirely fits in the produced image. The field of view is easily understandable to be highly dependent on the magnification of our microscope. The parameters represented here are related between each other, and it is indeed difficult to have the best of one of them without sacrificing the other 2. However, the application often defines the trade-off. As an example, if we aim to see subcellular structure in fixed cells, most probably we can push to have the best spatial resolution without caring about the temporal resolution, as in this sample, nothing is going to move. If instead we are interested in the movement of single cells in the development of an entire embryo, we probably have to sacrifice some spatial resolution in order to have a big field of view and an acceptable time resolution. Also, a common mistake by non-expert users is to think that the more we magnify the image, the more we will be able to solve the smallest feature of our specimen. This is not true, as while the magnification can be easily increased by sacrificing the field of view, there are physical limitations to the spatial resolution. Let's now have a look at the schematic of a simple microscope, in which we can observe some of the basic components. The light source, in this case a simple lamp, illuminates the sample of interest, and some of the light will travel toward the objective lens, while the rest of the light will be absorbed and scattered by the sample itself. This creates a shadow of the specimen that the objective lens delivers to the so-called tube lens. This light will be then projected toward the detector that will record the image, which finally is a measure of the light intensity levels across the observed objects. One of the main components of a microscope is the objective lens. If we think that each point of the sample emits light as a spherical wave, a certain portion of this light will be directed toward the objective. Its function is to collect this portion of light, which is a solid angle, and transfer it as a collimated beam to the tube lens. The broader the collected solid angle of light, the higher the number of photons that will contribute to the image-forming processes. This scattering ability is measured through the numerical aperture. Reversely, the numerical aperture also defines the focusing power of the objective, and therefore its ability to distinguish 2 points at the focal plane. For this reason, the numerical aperture of the objective normally defines the best spatial resolution achievable from the entire microscope. Commonly, increasing the refractive index of the medium between objective and specimen leads to an improvement of the numerical aperture and therefore of the spatial resolution. For this reason, one can find objective designs to work in different immersion medium, like air, water, or oil. The objective, however, do not only define the resolution of the system, but it largely contributes also to the field of view of the system through its design magnification. Moreover, at the interior of an objective there is a complicated set of different lenses that are designed to minimize the so-called optical aberrations that could give rise to imperfection and misleading artifacts in the final image. Therefore, it exists a high number of different microscope objectives, but again, the application often dictates a range of usable ones and therefore a range of possible spatial resolution and field of views. After the collection of the light coming from the sample, the objective transfers the photons through a collimated beam to the so-called tube lens. This is a high-quality lens that will refocus the light onto the detector to form the final image. The ratio between the focal length of the tube lens and the one of the objectives define the overall magnification of the system. Different brands of objectives use different tube lens focal lengths, and therefore the magnification specified from the objective is true only if used with the correct tube lens. This overall magnification can indirectly also contribute, in combination with the detector, to a variation of the spatial resolution with respect to the one reachable from the objective. So, as said, the tube lens refocuses the light onto the detector, which is placed at the so-called image plane. There exist different types of detector, the most common of which are the eyepiece lens and the user's eyes, the multiplier tube, which can record the incoming intensity without being able to spatially discriminate the source, and the camera, that can discriminate the source of the incoming light. In this lecture, we will concentrate only on cameras. So the main component of a camera is the so-called camera chip, an array of pixels that are capable to convert the incoming light intensity into voltage and finally into bits. This information is then sent to a computer, permitting to record, store, and process the image. For the camera, a number of parameters are defined. One of the most important is the quantum efficiency, that refers to the percentage of the incoming photons that are detected and converted into voltage, therefore contributing to the final image. The higher this percentage is, the brighter will be the image. However, beside the photons coming from the sample, also noise sources can be included in the signal at the output of the camera. Those can be of various nature, like optical as ambient light, or electrical from the camera amplifiers, but they degrade the overall quality of the image that can be measured through the signal-to-noise ratio. Other important parameters for the camera are the pixels number and their physical size. The number of pixels contribute to the field of view. The higher the number of pixels, the larger the possible field of view. The physical size of the pixel instead can contribute to the spatial resolution of the final image. In fact, if the pixel size is too big or the overall magnification too low, we may not be able to project different points of the sample into different pixels. Therefore, despite the fact that the objective could be able to collect photons from different sources, we may not be able to discriminate them in the final image. Another performance measure for cameras is the readout speed, or the minimum time interval between the capture of two consecutive images, that also contribute to the temporal resolution of the system. So, the aim of this lecture was to give an overview on light microscopy, its principal components and the main parameters. In the next lectures, we will concentrate on fluorescence microscopy. The working principle, briefly, consists in connecting a fluorophore to a particular feature of interest in our specimen. Then, by illuminating the sample with a specific color, we will force it to emit photons in another color. We then collect only the light of this specific wavelength, and by doing so, we drastically increase the contrast of our images. Here on the left is an example of fluorescent microscopy imaging, in which the membrane of all the cells of this zebrafish are labeled. As we will see in the next lectures, some microscopy techniques also permit to retrieve the position of the fluorophore in depth, enabling what is called optical sectioning. This permits to create a sequence of section of our specimen called Z-stack and allow its 3D reconstruction, as we can see on the right image. Thank you for your attention.